Welcome to this last but not least session of this fifth online Polymer Congress meeting. It's a great pleasure and privilege to have today with us Professor Dimitris Vlassopoulos. Professor Vlassopoulos is a professor at the Department of Material Science and Engineering of the University of Crete and an affiliated faculty member of the Foundation for Research and Technology in Crete. Dimitris is an expert in rheology. He has received the 2015 Weisenberg Award from the European Society of Rheology and the Bingham Medal in 2019 for his achievements in the field of experimental soft matter rheology. Today, he's going to talk to us about environmental conditions and pressure, how they affect the rheology of supramolecular polymers. Dimitri, thank you very much for accepting this invitation.
Thank you very much, Maria and Sergey and all organizers, and good morning or good afternoon to everybody, wherever you are. I very much appreciate the opportunity to discuss here some of our work on supramolecular polymers, and obviously this is only due because of our next speaker who introduced the field to the planet. But I'll take a different angle here. And so let me start by just mentioning the people who really did the work. So it's Manos Veroudakis, a former master's student, Nikos Burger, who is actually completing his PhD, and Benoit Lopinet, a close colleague of mine in Crete. But really, I would like to acknowledge my two chemist collaborators here, Laurent Bouteiller in Paris and Bert Meyer in Eindhoven, as well as, of course, Nathan Van Zee, his former postdoc, who is now at ESPCI in Paris, and of course, collaboration with Gerd Meyer in Jülich. And as I move along, you'll see where those important people come into play. So this is the menu for today. I'd like, first of all, to separate the view of these materials from the chemistry or physics point of view. I think that supramolecular polymers are coined by the chemist, and rightly so. Physicists tend to call them living polymers, but for us, it doesn't make any difference, but I will explain really the basics of the understanding of the viscoelastic response of these materials. And then I'm going to focus on such systems in apolar solvents, and in particular, we'll see how the hydrogen bonds, which keep the assemblies in place, is affected by different environments. One will be humidity and temperature, and the other will be pressure. So as a means of introduction, one can even think of a simple surfactant worm like micelles, where you have an amphiphile here, and under certain condition, it's going to really produce this interesting worm like micelle, the tube here, where the inner part is the sulfophobic and the outer part is the sulfophilic component. And then, of course, the chemistry is always ahead of physics. And then there are different motifs that you can see here schematically that allow to make large self-assemblies, typically in the form of fiber-like, or if you want, flexible-like chain and so on. But here you can see more detail the different types of interactions that you have. In this particular case, you can have both hydrogen bonding as well as pi-pi stacking interactions. So now, what is characteristic in all the systems from the standpoint of our interest in understanding the physical properties is the large distribution of lengths, which depends on concentration. So here, this is an example where we vary the concentration of the added motifs in a particular solvent, and you see how the distribution of lengths changes. And here, this is the average length that depends on concentration. And so this here, Boltzmann factor, has as characteristic the apparent activation energy of the bond. Right. So then these are systems which are a priori polydisperse. Now, the main contribution in terms of understanding the viscoelastic response of these materials is due to Cates almost 40 years ago or 35 years ago. This is simply summarized as follows. There are two characteristic net scales in those materials. Think of them as a simple worm-like chain. So then this is the so-called break time, which is the association, the time of the bond that keeps the worm-like chain intact. And then there is the long relaxation time of the whole chain. So now in case the break time is very long, so that means that the whole chain stays intact. You have the classic Dejean approach of raptation that you can see here schematically. So the thing stays in a tube in a large concentration where you form physical networks, and then the escape of the tube that you can see schematically here, and I kept the cartoons from the original Cates paper, basically dictate the time. So how can we check the time? This is the moduli as function of frequency in a simple oscillatory measurement. And so the dark line is the storage modulus, or else reflecting the elasticity of the material. And the dashed line is the loss modulus, or else reflecting the viscous dissipation. So now the crossover here, it basically marks the onset of the flow of the complete liquid-like response of the material. But one thing to note here is that this G double prime, the viscous dissipation, is extremely broad. And so this broadness here reflects the distribution of length that I mentioned before. So this is the case, again, of a very large break time. So now, in the case of a slow break time, of a fast break time, so in other words, when the system is dynamic and it breaks and reforms, and so you can see this in the bottom cartoon here, where the tube here will break and then it will reform. So then the overall response, as one may expect, is faster. You still have the same plateau here, but the interesting thing is this is now faster because the crossover occurs at higher frequency, therefore at shorter relaxation time. But the interesting thing is that now the peak here of G double prime is much more narrow, and actually is characterized by, or it's described by a simple Maxwell mode, so a simple exponential relaxation, right? And this is the reflection of this redistribution of lengths because of the fact that the break time is short. Okay, so then the main thing now is we're going to use this, and we're going to first look at supramolecular assemblies in a polar solvent. So then, as I mentioned, they're based on hydrogen bonding motifs, and we're going to see how humidity and temperature affect the viscoelastic response. Why are we interested in this? Well, when we go back to the theory of Cates, right? So this is basically, in this particular case, what the theory predicts. So again, this is the case for a short break time, and there are predictions for the modulus. This is the plateau modulus, or the modulus of the physical network form. This is the terminal relaxation time, and the product of the two will give us the so-called zero shear viscosity. So then the prediction for the modulus is such that there is a power law here with an exponent of about 2.25. And for those in the field will recognize, for any polymeric network, you have basically the same scaling exponent. The terminal time, however, has a scaling exponent of 1.25. When you see the available data in the literature, again, for systems which are characterized by hydrogen bonding assemblies in polar solvents, okay, the scaling exponent goes all over the place. It can even be negative. So then the question is, what's going on? Which is the correct experimental exponent? So this simple question we've tried to address first with collaborating with Laurent Bouteiller, who is an expert, among other things, in this IHAT motif. So this is ethyl exyl urido toluene, okay, which forms different kinds of self-assemblies depending on temperature and concentration. The simpler one is called filament, and so you have this arrangement that you can see here in the cartoon, whereas when you have a tube-like arrangement, where basically you can take a cross-section and you have in each cross-section 3 of these motifs here, this occurs at different range of concentration and temperature. How one can basically check this, for instance, rheologically, okay, which is the tool we many use. Okay, we take this IHAT and we dissolve it in dodecan, right, which is clearly a polar solvent. This is a particular concentration. And so this is a busy plot. Let me walk you through. We still have here moduli as function of frequency, and the measurements are done either with conventional rheology or with passive microrheology that I will explain later on. But at the moment, we take it for granted that it can really provide moduli. And so you have data all over the place. So let's look only at the dark lines, and the dark lines are the G prime, so in other words, the storage modulus. So you have at very high temperatures this kind of behavior, and this here slope is about 2, which is a classic viscoelastic liquid. So then the behavior changes as the temperature is reduced. It changes further as the temperature is further reduced, and so on and so on. And so then with reducing temperature, the overall picture here, based on what we know from conventional polymers, goes to unentangled filaments, to unentangled tubes, to partially entangled tubes, and to well-entangled tubes. Okay, so there is a whole range of different systems here. And so by doing this experiment for different concentrations and temperatures, one can construct a phase diagram like this, right? And so you see the different regimes, very high temperatures, you have unentangled filaments. And so then depending also on concentration, you have unentangled tubes, partially entangled tubes, and well-entangled tubes. And basically the lines here separate the different regimes, but basically are almost iso volume fraction C over C star, where the C star here, okay, the C over C star depends on the activation energy, and the activation energy depends on the concentration, and this parameter is also a value of concentration. So the overall analysis here provides an activation energy which is consistent with what one expects for these kinds of motifs from the literature. The point here to stress is that you have a phase diagram, so you choose the temperature and concentration, you know exactly where you are. Now, the reason why this is important is because if you go back here to viscoelasticity, here you have predominantly liquid-like behavior, here you have predominantly viscoelastic behavior, and for a wide range of temperature of frequencies, solid-like behavior. Now, these data are here scaled also with the changing viscosity of the solvent at different temperatures. Okay, so now, if one looks at more detail at what I mentioned before, this is just one linear viscoelastic spectrum, and the terminal regime near the crossover toward the terminal flow is well characterized by the red lines, which are a simple single exponential decay, which is the Maxwell model. So now a question is how stable the structures are over time. And so here I show you such a spectrum for a particular concentration and temperature. That's the first test. And then after a few hours, you take a second test and you see that things change. And note that you may say that this is not much change, but this is a double log scale. So the change is certainly significant. Okay. And so this is experiment done in the open air, and the open air has, of course, humidity. And of course, the key question now is whether humidity plays a role. And it turns out that in the literature it is known that even 10 minus 2% water may be dissolved in alkanes. So then it's basically molecular scale dissolution. And so that prompted us to really control the humidity in a better way. And this is a simple, basically a sketch of a setup we have in the lab. It's extremely simple. So this here is the cone of a standard rheometer that it's used to really measure. And basically the sample it's here in this area. And so then we kind of form this chamber where we kind of close the whole setup, and we introduce a salt, which is hygroscopic. And so also we have a solvent. So then we create an atmosphere which is saturated with solvent that at the same time absorbs all water. So typically this is one of the salts we use, phosphor oxide. And so then knowing that how it's hydrolyzed, we can estimate how much we need. And of course, we have an excess, so we make sure that we control the humidity well. This is an example of, you can measure, of course, because we introduce a humidity meter, right? And so then you can control the humidity depending on the salt you have over a long time, and it's quite constant, and we can go 5% and even a little bit below. So with this in mind, now we can do measurements at different levels of humidity. So to show how important this is, I compare here two sets of data with the same system. The first one, which is now with a high frequency crossover, so very fast response, is the one where you are in open air, so in the presence of 100% humidity, more or less. Whereas if you reduce the humidity dramatically with this drying chamber to about 5%, you see that you have almost the same plateau, almost, not exactly, right? But you have a dramatic decay here of the frequency, crossover frequency. So then humidity does play a role, and in a way, in this particular system, it speeds up the relaxation time, whereas it affects a little the plateau modulus. So the idea that came up not from us, we just tested it, is that the water acts as change stopper, and this cartoon here shows pretty much what's happening. This is different concentrations of our motif. The motif is the hat. So then when you add water here, which is this red ball here, is going to basically stop the growth of this chain in this direction or the growth of this chain in that direction and so on. So basically it's going either to break or stop the growth of self-assemblies, right? And so the way people understand this is that you have basically the effective supramolecular polymerization by adding a motif to an existing chain, okay? And so then if you add the stopper here, chain stopper, that's called, which is the water, you have this formation here of this effective complex. And so then you can determine the degree of polymerization in this way. And so then you can have results of this form, the effective degree of polymerization of the grown micelle as function of the concentration. And so if you don't have a stopper at all water in the absence of humidity, you follow this straight line. But then for different level of humidity, you have this kind of behavior. So now one can basically take the assumption that the time is proportional to the length scale, and therefore to the degree of polymerization. And so then the data that I showed you before are basically this red data here. And so then we can see that they are between these 2 degrees of polymerization, okay, of the chain. And so this here will correspond to about 10 ppm of water in the particular system under the present experimental condition. And the independent calcium titration yields 25 ppm. So I think all things considered, we are basically on the right track. The important thing is that you can control really the effect of humidity, which does have a significant effect on the viscoelastic behavior. And now with this in mind, one goes back now to the scaling. And so this is the excuse me, the plateau modulus as function of concentration. And here I'll show you our data, which are the pink ones, as well as data from the literature. And as also it was shown before, the humidity has very little effect. And so pretty much the scaling remains consistent also with the theoretical prediction. But let us now see what's going on with the relaxation time. This is our data here, which show that there's a scaling exponent of about 3/4, which is smaller than the theoretical prediction. We have some ideas about it, but we're not sure. And this is the level where the things stand right now. But it's very different from other literature reports where the scaling reported was very different. And we do believe that this reflects clearly the effects of humidity. So our experiments have, I would say, practically no change stopper in the system. Okay, so now this is just a little on the side information to say that this affects not only the linear response, but also the nonlinear response. For example, what I showed you here is when we are trying to stretch this network and to see how it responds to strong forces. This is what we call the transient viscosity for different applied shear rates. So basically rates of deformation. And what you see here is for each color, 2 different types of data, lines and symbols, and the symbols correspond to the case of humidity-free response. So qualitatively, there is not much difference, but quantitatively, there is dramatic difference. And this kind of increase here is basically the stretching of the network that one can see here, that one can describe, I would say, phenomenologically. So then, that means that there is an effect of both linear and nonlinear response. And then we decided to team up with Bert and Nathan that I mentioned before, because they work extensively with this other hydrogen bonding motif, this biphenyl tetracarboxamide, okay, BPTA, that you see here. And they have independently discovered that water plays a role, but also in conjunction with temperature. And so basically in this case, they didn't talk about chain stopper, but they talk about copolymerization. So then in a way, at high temperatures, there is no effect, which is something that one may expect, because obviously at high temperatures, water may disappear, but at lower temperatures, water will intercalate into the self-assembly and form this kind of effective copolymer, which may be different and has different microstructural characteristics as they determined from circular dichroism studies. So now what we did is we also worked with this system, and we look first at the static response. So then we did static light scattering, and indeed that there's 3 temperatures which correspond to 3 different types of configurations. Here we have some difference in the conformation. Of course, the data are not sufficient because with light scattering we could not go to lower Q, but clearly at the lower temperature, we seem to have something which is more consistent with flexible or semi-flexible chain, while the point 2 here is actually consistent with Gaussian chain, whereas the -1 here is consistent with more rod-like polymers. So then the conformation really changes with temperature and effectively with humidity. So now these results will suggest also from analysis that you have a large persistence length and a very large contour length, which is consistent also with independent electron microscopy studies. So now this is a bunch of data at different times, and you see that now there is a strong time dependence. And so here you see the start and the end of the experiment. So then at very long times here, you have a dramatic slowdown. And so again, here you see that actually this is a new phenomenon, that there is a large effect of time in the system. So accounting for this effective aging here, you can basically measure data of the age systems at different temperatures, and you see that at low temperatures you have a behavior which is consistent of a viscoelastic liquid, and as the temperature increases, you form a more extended plateau and you go towards something which is more viscoelastic liquid, but highly viscoelastic material. You can do the same experiment by changing the level of humidity, and you start from the high temperature and then you introduce more and more humidity. And so basically, as we saw in the previous case, you have effectively liquefied your material, so you weaken its viscoelastic behavior. So with this in mind, one can summarize all the data. And so we look at the scaling of plateau. This is the different BPTA systems. So this is the plateau modulus as function of concentration for the 3 temperatures. It's the same. I can add here also the HAT system, and the scaling of K's is pretty much respected. So now I go to the time. So this is the relaxation times. This is the HAT system, and this is the 2 different BPTAs at the lower concentrations. At the low temperatures, the more the less flexible, if you wish, whereas the more whereas the more flexible has a completely different scaling. And this scaling goes kind of in the direction of flexible chains. So now we are always at atmospheric pressure, and so a summary here of effects is that with a HAT, there is no aging at all, whereas ours already indicated there is a significant aging of BPTA. So then the specific type of the motif plays a big role in the viscoelastic response and in the equilibration of the system. Now, one can then look at different levels of humidity, the relaxation time versus temperature in the age systems, right? And one can see that with a HAT, you have fairly similar activation energy, which is the slope here, whereas this is not exactly the case, or is much more unclear with the BPTA, right? So this suggests that, again, the activation energy for flow in the material is very different, but this is now under different levels of humidity, right? And the humidity affects activation energy, and certainly the time, much more in the BPTA system. Okay, that brings me to the end of the first part, and I realize that I have about 5 to 6 minutes more, okay? I guess, Maria. So then humidity is critically important in affecting the, the rheology. I've just mentioned that there are perspectives in the nonlinear response, and there is an open window here of opportunity to exploit the role of these coupled interactions, and maybe solution processing can be facilitated in the presence of water. So let me quickly now go to the second part, which is the effects of pressure. We started looking at this thanks to our collaboration with my longtime friend Gerd Meyer from Fortress Jülich in Germany. And so what we do here is a simple microrheology experiment. So then this is a light scattering cell. You have your material, and so you send a light, and then you probe the scattering intensity. So now you have some probes, which are some simple particles. And I can discuss details if somebody's interested. And so then you get basically the intermediate scattering function. So now you can independently, of course, add an inert gas here. It can be nitrogen in our case, or it can be argon with a compressor under controlled pressure, right? You make sure that the diffusion of the gas in the liquid is slow enough so that you can measure properly at the measurement volume at the particular position where you are. When you do so, you can basically extract the mean square displacement, which is a function of the compliance here, which is inversely proportional to the modulus. And so then through Fourier transformation, you can get the viscoelastic spectrum that you can see here. And so just to show you that things work well, the entire curve is due to this passive microrheology, whereas the blue data are just the data you get by conventional rheology. And the analysis of this here, the slope here, gives you the opportunity to appreciate the and actually estimate the persistence length. So with this in mind, we can now look at the effects of pressure, right? And so here you see that we vary the pressure by 3 decades, and we go again from unentangled filaments to partially and well-entangled tubes. And so then for a particular concentration, you have a phase diagram now, which is temperature versus pressure. And you see in this particular case, which you see half, the pressure tends to stabilize the tube phase here. And so this line here, we can understanding as a Clapeyron as a first order transition. Simply, I'm not sure if it is, but that's the only thing I know from thermo. But if you use the Clapeyron equation here, because you have all the known here, and the delta H is known for the particular motif, you can estimate a change of volume of your motif, which is about 8 cubic angstrom per molecule. And this is pretty much what simulations show. Okay, so that means that somehow we have a feeling about how the pressure acts on their systems. So now it turns out that BPTA pressure is different, and this is my one but last slide. So now the pressure here seems to weaken the viscoelasticity of the material at low temperature. But if you increase the temperature, the pressure has actually stabilizing. Right? So then the interplay of pressure with the motif is intimately related, of course, to the activation energy, but as well as to the ability of the motif to really change its relaxation time, its break time, of course, with temperature. So this is, I would say, it's in view of time, I will show you the summary of this data here. So that's the relaxation time. So BPTA, as function of pressure, can either lower or almost be independent, whereas with a HAT, it speeds up. So it's very different effect. And that's here are the conclusions. So we do have the ability to measure the rheology at very high pressures. This is interesting because this is a thermodynamic parameter partially overlooked because it's not so easy to do the measurement, but the pressure here can basically play an effective role equivalent to humidity. It can do whatever you want. It can stabilize a phase, it can promote a phase, it can basically reduce the viscoelasticity of the material. And of course, this is a complex interplay that still needs to be better understood. So on this happy note, I'd like to thank again my collaborators, and in particular people in the first row, especially the 2 students who did most of the work, eUseMe, which is a platform that helped the financing the project, and you for your attention.
Thank you very much, Dimitris.